Hello everyone, a very happy new year to all of you, and I wish in this year you will get all the happiness and success in your life. And in this 24th video of PYQ series, I'll be discussing about Albert Einstein. So without wasting any time, let's start with the video. So the MCQ asked was a patient present with complaints of fever with chills. His throat examination revealed swollen glands and a thick white membrane over his tonsils. Albert Einstein is to be done. Which of the following is constituents of Albert Einstein? So we'll see the options. Carbol, fuchsin and malachite green. So if you remember carbol, fuchsin, malachite green, these two are the reagent for Zn stain. Okay, Zn stain, which is done for Microbiology tuberculosis and other acid-fast bacteria and parasite. Okay, so we will discuss about Zn stain in our next video. Okay, so this first one, carbol, fuchsin, is the primary stain in that, and malachite green is counter stain. Second option, so this is out. Second option, carbol, fuchsin and methylene blue. Again, these two are the reagent which is used again in Zn stain. But then instead of malachite green, methylene blue is used as counter stain. However, the primary stain is same. So this is also out. The third is toluidine blue and methylene blue. Toluidine and methylene blue, though these two reagents together used in Pondus stain, which is used to stain the metachromatic granules of Corynebacterium diphtheriae. Okay, but this is not Albert stain. So this is Pond stain. So this is also out. Now the fourth option, toluidine blue and malachite green. These two are the reagents of Albert stain. But then Albert stain is composed of two parts, solution A and solution B, or solution 1 or solution 2. So these two reagents are part of solution 1 or solution A. Okay. Remember this. So this is the correct answer. So we will see the stain. The Albert stain is what type of stain? It is a special stain. Okay, it is a special stain. And why it is called a special stain? Because it is used especially to stain a particular characteristic of the bacteria. And what is that? That is the volutin granule present in the Corynebacterium diphtheriae. And these volutin granules are basically strongly basophilic bodies, and it is composed of inorganic polyphosphates or polymetaphosphates, also known as poly-P. Okay, polyphosphate or polymetaphosphates, also known as poly-P. And these are the storehouse of the cell, and that's why it is, this granule is also known as storage granules. Okay, storage granules. So what are the reagent which is used in Albert stain? It composed of solution 1 and solution 2, as I told you. Solution 1 is composed of toluidine blue, malachite green, glacial acetic acid and alcohol. And initially when Albert stain was designed, instead of malachite green, methyl green was used. Later on, Le Bon has given a modification and replaced the methyl green with malachite green because it gives a better staining to the metachromatic granules or the volutin granules, and thus after that the Albert stain is known as Albert's Le Bon method. And the Albert solution 2 is basically Albert's iodine. Albert's iodine, as we have seen in case of Gram's stain, Gram's iodine. So it is composed of what? Iodine and potassium iodide. Okay, so what are the steps for staining? It is very simple. It is two-step procedure. In first step, we'll add the Albert stain 1 or Albert stain A, and we'll leave it for 5 to 7 minutes. Later on, we'll pour off the stain and wash the slide with Albert solution 2, that is Albert's iodine. And then we will apply the Albert's iodine for 2 minutes, and then we will wash it with water and blot dry it and examine under microscope. And what we'll get? We'll get this result. So what we can see in this image? We can see the darkly stained granules. Okay, so the granules will take up the bluish black color or bluish purple color, and the bacteria or the protoplasm will take up the green color of the stain, and the other organism is present, it will take up the light green color. So these volutin granules, if you see, are present in the poles. Okay, are present in the poles, and that's why it is also known as polar bodies. Instead of polar bodies, they can also be known as Babes-Ernst granules. Okay, and they are also known as metachromatic granules. So now we know the four name of these volutin granules: polar bodies, Babes-Ernst granules, metachromatic granules and storage granules. Why it is known as metachromatic granule? Because of its ability of metachromasia. That means if you stain it with a certain dye, it will take up another color different to the color of the dye used, as in case of Loeffler's methionine blue, in which the color of the dye is blue. Okay, but what color it will take? It will take bluish purple. Okay, that is why it is also known as metachromatic granules. Okay, so you can see this bluish purple granules present at the end of the or the pole of the bacteria. Okay, what other stain which can be used to stain the metachromatic granules? The nasal stain, which composed of three solutions: solution A, solution B, and solution C. Solution A is methylene blue, solution B is neutral red, and solution C is grams iodine. There is a modification to it in which the solution B, neutral red, is replaced with crystal violet, and solution C, grams iodine, is replaced with chrysoiodine. And the color what the metachromatic granules will take up, it will take the reddish purple color. Okay, so now you know why we call it metachromatic granules, because it will take a different color to the color of the dye used. Pond's stain, as I told you, it composed of toluidine blue and methylene blue, and it can also be gram stain. But then gram stain will the granules will take up a strongly positive gram stain compared to the bacteria, but then this gram stain tend to decolorize easily. So that's why we don't prefer gram stain for staining the Corynebacterium diphtheriae. For identification, we use Albert stain, or we can also use nasal stains, okay, but not the gram stain. So I hope this short video will help you to answer all the questions related to this topic. So just go through it and make your own notes and revise it. Take care. All the best.